Awesome. It's so great to see so many familiar faces and new faces. Thank you for joining us today. I know it's really early for some of you. I know it's late for some of you. It's hard to find a good time when we have such a global community, but thank you for being willing to make time in your schedules to join us to talk today about inheritance and mosaicism and SCN8A. This is a topic that we've been thinking about a lot in the last couple of years, but haven't really been able to dig into quite as much because we just didn't have enough families. Now we're really starting to see families come in. We have families who are stepping up and wanting to be a part of the research and share their stories. And that is what is going to drive us forward. So thank you all for being here, for sharing your stories, for being willing to open up, because it's not always the easiest thing to do. This is a pretty intense thing in our lives to deal with. But we are learning so much from all of you, and we're really grateful to have you today. If you don't know me, there's some faces that are new on here or names that are new. My name is Gabi Conacher. I am co-founder and executive director of the International SCN8A Alliance. Michael Hammer and Jayetta Hecker, who is my mother, are my co-founders. And we've been working together as a team, Michael, Jayetta, and I, since 2014 on trying to find better answers for those living with SCN8A. And I'd have to share that I feel like the tide is turning. We are at such an incredible inflection point for SCN8A. We know more today than we've ever known before, obviously, because we're learning from all of you. But SCN8A is out there. SCN8A is known. More and more known. Obviously, it's still, a rare disease. But I think if a lot of the meetings that we're going to, we go to a lot of meetings with researchers and clinicians. SCN8A is something that more and more people are seeing and learning about. And I think what's really driving that is the registry, families sharing their stories, sharing their experiences. This year, out of the SCN8A the International SCN8A registry, we are going to have at least 4 papers that are going to be published. So that is what's driving the knowledge that is out in the community and that we're learning from. And I'm sure not too far behind are going to be some papers on inheritance and SCN8A. So today, that's really what we're going to dive into. Quickly, I'll share a quick update. I think many of you know that we have diagnosis and treatment guidelines that are in process, which is the first global effort to really gather what we know about SCN8A and put it in one place so that we can essentially have treatment guidelines for our children. We know that our kids are very, very different, right? My Elliot is very different from, a lot of other kids. Megan's daughter, Chloe, is her own unique story. Everybody's kid is unique in their own ways, but we are learning some things about what the best kinds of treatments are, the best possible care that our kids can get. And from this process, which has been going on for almost 2 years, we will later this year, we hope, print these first treatment guidelines, which means that no matter where anybody lives or who their doctor is, they can find these guidelines to help their kids get the best possible care and treatment as soon as possible. So we're really, really excited about that. That's moving along. And that's being done in conjunction with more than 30 pediatric epileptologists from 5 different continents and 13 different family caregivers. So it's really a community effort there. So I don't want to take up too much more time, but I know people have that question. I will give one more quick update on clinical trials. I know that's on a lot of people's minds. We know that if people are interested in learning more about the trials, we have it up on our website. I will put the link in the chat while we're going. But essentially, both clinical trials, neither of them are recruiting right now, but there is a pre-screen going on for the Praxis 562 trial. So if you're interested in learning, if you're eligible, there's a nurse that you can speak with. You can set up time to speak with, and they'll help you understand. And even if you're not eligible now, some of the criteria might change. So if you are interested, I encourage you to call. It is just US right now, but soon will be opening up to other countries. And then the Neurocrine trial is not yet recruiting again, but they will soon, in the next probably 6 months, be opening up to to bring another 52 patients into the trial. There are currently 8 that are in the Sentinel, is what it's called, the first 8 where they gather a lot of data, do the data processing, learn from them, and then make adaptations for the open trial. You can also, I'll put the link to the links to on that for both of these are on our website, and you can learn more. So I don't want to take too much more time. I'm going to hand it over to you, Michael. And just as we're talking, feel free to throw questions into the chat. I will be monitoring that while things are going on, even if it has nothing to do with the topic at hand. If it has something to do with SCN8A or resources or community, please, we're here to support you.
All right. Well, hello, everyone. How are we doing on languages? Are we getting our translation thing working yet?
It's not working.
Okay.
I posted that it wasn't going to work. Unfortunately, the setup was having some trouble. So, but I think we can do this again someday and have it be translated.
Okay.
So I
All right.
apologize to anyone who wasn't looking forward to that. We were too.
Okay. So if there are questions, please put your hand up or put it in the chat. Elise will be monitoring and Gabi will be monitoring for questions. I want to introduce Elise Tanzer to everyone. She's our research clinical coordinator. And I know you've been getting emails from her, and a lot of you have been in touch with her directly. And she's a key person in the conduit between families and myself and Gabi as well, because she makes infographics. She helps us get the data cleaned from the registry. She helps you get the data from your families into the registry, and it's just making everything work so much more nicely. So we're very happy to have her with us, and I think this might be one of her first meetings where she's joined the families. No, okay, I'm not speaking accurately here, but it's one of the first. All right, so what I want to do, and like I said, it's very informal, but I'm going to go through some of the science of inheritance and mosaicism. And then really what I want to do is hear from you all, especially families who have one or the other or both. You'll see they're not mutually exclusive. So I will start and share my screen. So today's talk is on inherited mosaics. And this is a complex slide kind of summarizing de novo and inherited and mosaic variants. And I'm going to go through all of this in detail. If you want to go back to the recorded session we had last week on DNA mutation and inheritance, I covered some of this in that talk, and it's recorded and it's available online. And that one is available broadly in that, right, Gabi? So Gabi can put the link for that perhaps in the chat. I don't recommend you watch it while I'm talking, but I did cover the basics on some of the things I'm going to go over here.
Everyone should have also received an email about yesterday announcing that this meeting would be today that included a link directly to that basic science talk as well.
Great. So I'm going to cover a little bit of back review here, and that is what is a genetic mutation and when does it happen in your body? And this is all very important for understanding inheritance and mosaicism. So the genome, which is the sum total of all the DNA in any given cell, is made up of 6 billion nucleotides. And nucleotides are the A, G's, T's, and C's on the strand of DNA. And sometimes they're just referred to as A, G, T, or C. Sometimes they're called bases. Sometimes they're called nucleotides. But the point is, every time a cell divides, the entire complement of your DNA has to divide, make a copy of itself, and then find its way into the daughter cells. So starting from, for example, when mom and dad's sperm and egg meet and form a zygote, immediately that one egg sperm combination starts to divide into 2, into 4, into 8, into 16. And it goes through various stages, as you remember from your high school biology, eventually forming the organism. but that process is just cell division after cell division. And in doing so, like I said, the entire complement of the DNA has to replicate itself. It's called replication. And here you can see the two strands of the double helix unwinding, and new strands are being copied and made on the parent strands. So the daughter strands, the parent strands. And there's a lot of room for error because there's 6 billion nucleotides, and any single copying event can make a mistake, but it's very, very accurate. So in the course of dividing, very few mistakes are made. And so your cells in your body pretty much are all identical. However, some may have a mutation. And if it's in your arm, your leg, your heart, your brain, it's called a somatic mutation. If it's in the cells in your germline, the cells that make sperm and egg, it's a germline mutation. And we're going to go through that in detail. The point is that every one of us inherited about 50 to 100 novel mutations that our parents didn't have. So in the extreme case, you might have a couple hundred of these. In the more less extreme case, maybe only 50, but everyone has this number of new mutations. And the point is, those mutations can happen anywhere in the genome, in any gene or in any piece of the genome that's not coding for a gene, and it may have no effect on the overall phenotype of the person. It may not affect any trait or a disease, but occasionally, with bad luck, one of these mutations will wind up in an important gene like SCN8A. And that's what brings us all together here. So I just want to make it clear that there's nothing special about getting a mutation in a gene that your parents didn't have or your parents did have. It's happening all the time as part of nature. It's the natural process. Any questions on that? Okay. So as I alluded to before, mutations can happen at various times in our development. And there's two genetic cellular processes that are different. One is called mitosis, and that's the process of making new cells for your body, anywhere in your body, like just cells dividing to produce skin cells as they differentiate from the embryo into heart, lung, liver, brain, skin. That's just cell division and differentiation from a stem cell to a cell that has a very specific function. And that process, as I described previously, is ongoing, and the DNA is replicating, and in that process, mutations can occur. That's over on this side. In the germline, there's something called meiosis. And the reason why you need a different cell division process in the germline is when you make a sperm or an egg, you need half the DNA complement, so that when it merges with, a sperm merges with an egg, it recreates the full complement. So I didn't mention this before, but you can go see it on the talk from last week, that the DNA is housed on these things called chromosomes. And we have 46 chromosomes, but we get 23 from mom and 23 from dad. And so in the process of meiosis, those chromosomes break down into sperm or egg cells that have only 23 chromosomes. And when they merge to form the zygote, they recreate the full complement of DNA that you expect to have in your normal cells. If there's a problem and an extra chromosome gets moved into a sperm or an egg, and that ends up in the individual, you get a trisomy. Like Down syndrome is an extra chromosome 21. That's a mutational process that can occur as well. But we're going to focus more on the kinds of changes that affect just a single spot in the DNA strand. We call it a point mutation, or a substitution of one nucleotide for the other. I don't have a slide showing the P dot and the C dot. That was something I covered last week. But if the mutation will occur in the DNA, and it will end up expressing itself in the protein. The protein version is P dot and the DNA version is C dot. All right, now let's look at the question of how mutations are inherited. So there's three cases I'm going to highlight. The first is an inherited mutation. This is a mutation that is in an ancestor of some sort. It could be your mom or father, it could be their parents or their parents. And this, like I said, is a natural process. Each of us gets 100 new mutations, but most of those don't make a difference. And so they stay in the population because individuals can have normal lives, they can grow up, they can have families, and they can transmit those variations to the next generation. So in the case that one of those variants is very strongly affecting the individual, it will not be passed on because that individual will either not survive to have children or not be able to have children for various reasons. So that mutation will not be found in the population for very long. It will be weeded out. The de novo case is when a mutation arises in the immediate ancestor, father or mother. In this case, we're showing a single sperm is received the mutation in the process of meiosis, and that particular sperm is the one that fertilizes the egg and forms the zygote and the embryo and the individual child. So the father has the mutation in a single sperm or the mother in an egg and transmits it to the child. The mutation is detectable in all tissues of the child, but not in the parent. So when you get your genetic testing, the child will have the genetic variant in all of their cells, whereas neither of the parents will have the variant. That's known as a de novo germline mutation. But there's another way you can inherit a variant from a parent, and that is the parental germline mutation that we're going to call a mosaic. And that is, in this case, the father in his development, before he was a grown-up, when he was a little infant, an embryo, there was a mutation that occurred mitotically in the cells that gave rise to his germline. And it could be that many of the cells in his germline, and therefore many of the sperm that he produced, could have the mutation. But he may not have the mutation in the rest of his body. So he's a mosaic. A mosaic is that combination of two or more types that are in the same individual. And so that could be something you don't know in life. And you find out when you have a child that has one of the variants that was formed in that germline mosaic cell line. And the thing about that is the father has the germline mosaic mutation in all of his gametes and transmits it to the child. And the mutation is detectable in all tissues of the child, not all tissues of the parent. But the point is that that father could have another child with the same variant. So some families will have two children with the same variant, not twins, but successively have two children with the same variant. And that is likely due to this process of germline mosaicism. It may look like a de novo if you have one child, or if you have two children, it'll sometimes look like a de novo because the father tests negative. And again, this could also happen in the mother's case as well. So I'm going to emphasize the process one more time here, showing you what happens in the father's, the parental gametes. This is the father here. Somebody had a mutation at some point, either mother or their father. They fertilized the egg, it became an embryo. And in this case, the mutation occurs only in half the gametes. So notice that The mutation is in all of the cells of the father, but because of meiosis, half of his sperm have the mutation and the other half don't. So if it's a de novo mutation, a germline mutation, then half of the gametes will carry the mutation. If it's a germline mosaic, you don't know how many of the sperm carry the mutation. If it's a somatic mutation, none of the gametes carry the mutation. So in this case, the somatic mutation occurred away from the germline. And then in that case, you don't know that somebody has the SCN8A variant because it's in some part of their body that doesn't affect the brain, or it could affect the brain, but it's not transmitted. So that's what I wanted to say about how mutations arise, when they arise, and how they're passed on. Now I just want to say a few things about the phenotype of the parent.
Michael, can I just say something while you're going? So Elise is also putting some definitions in the chat while Michael is going, just because there's a lot of terms that are hard to, if you don't know science. So keep your eye on the chat or raise your hand. Maybe, is there any clarifying questions that people have right now? Anything that wasn't clear before we move on?
Also, if you have any words that don't make sense and you're like, I don't understand what it is, you can just put it in the chat and I'll define it for you.
Okay. So the somatic mosaic parent is typically unaffected or mildly affected. So we have some parents that are mosaics and they're usually unaffected, but it's possible to be mildly affected if the brain is affected by the somatic mutation. If the pathogenic variant is present in the sperm or egg, it can be passed on to a child during pregnancy. That's again, that's the mosaic parent. The child has has the pathogenic variant in all of its cells and it's affected. So it has the disease, but it's not a mosaic anymore because he has the cells, or she has all the mutation in all of their cells. So the mosaicism is not inherited. It arises in one individual because sometime during the development of that individual, there was a somatic mutation. It may affect the germline or it may not, but if it affects the germline and it's transmitted to a child, all of the child cells will have that variant. And that child, if they have a child, will not pass on. It will not be a mosaic. So all of its children will have it. If the pathogenic variant is present in the sperm or egg cells of the mosaic parent, there's a chance that future pregnancies may be affected. That's what I'm saying, is that all or some of the sperm or all or some of the eggs in a mosaic, germline mosaic, may be affected by the mutation and therefore can be passed on more than once. So if we just go back to the situation we are focusing on, there's inherited mutations, and that's the topic today. And there's parental germline mutations. That's also the topic today, because you could have an inherited variant that came from a mosaic parent. So these are the two things we're focusing on today. For the rest of the community, where variants are de novo and they only happen once, we're not going to consider that today. That's what we usually talk about with families. But today we're going to focus on these two cases. So let me make sure that's clear to everyone.
We've got a couple of hands. Laura, did you want to ask your question? And then Tammy can go after. Let's see if I can find you, Laura. I can. Here, you're muted. Sorry, Laura.
Sorry. Okay, so I wanted to know if there is currently a way to determine which of these scenarios is actually what happened. My daughter's geneticist said, oh, you don't have to worry about your son passing this on because this did not come from either parent. This is something that happened in your daughter's development. And I see that that is an option, obviously, based on these. However, there is also some other options here that it could have come from either or myself or my husband. And being that it can come from different tissue sites and that we did blood tests specifically, is this on my paperwork that I'm not seeing, or is this just not a known, we can't test this yet?
That's a really good question, Laura. So they're not going to tell you on your genetic test, because to determine whether you're a mosaic or not is a complicated test. And I'm going to talk about that in a minute. I'm going to talk about the genetic counseling implications of this.
Okay.
But in your case, if your son did not inherit your daughter's mutation, the same mutation your daughter has, then he's not going to pass on anything to any of his children.
So he has to get tested? Is that
I
what I
think-
understand?
I think you could have him tested for sure. If he's testing negative for, your daughter has 16, 17, right?
Uh-huh.
She's affected either because your husband or you had a de novo mutation in the sperm or the egg and it transmitted to her. It did not transmit to her brother. If one of you is a mosaic, then it could have transmitted to both your children. But since it didn't, he's free. He's scot-free. He can't transmit it.
Because he doesn't show any phenotypes, then he is definitely scot-free.
Yes. Well, you could have him tested just to make sure he doesn't have it and is for some reason not expressing it, which I
Yeah,
don't.
that's what I was worried about.
Yeah. And so we have families on the call today who are going to talk about how they got their other family members tested. And I think that would be a really important topic for today. I want to make sure that we thoroughly discuss that, because the clinicians who order these tests need to know when it's important to test a sibling. Okay. That's like a major, major thing.
Yeah, because my son just told me he wants 4 children, so.
The safest bet is that it's a de novo mutation and he did not get it because it just happened once. If it happened in a germline case and one of you is mosaic, then he's still scot-free because he didn't get it in the mosaic case either. If you have another child, then it would be possible to have the third child have it. And that's what the other genetic counseling implications are. When should we ask that we are looked at to see if we're mosaics? And it's a tough call because it's not a simple test.
Right, and the fact that I didn't continue to have children, I wouldn't know if I would have that third child, right?
Yeah,
That would possibly have it as well. So it's still a question of de novo or mosaic, possibly.
exactly. But because mosaic is not inherited, your son's not a mosaic, and your son doesn't.
I get it. Okay, yeah, I missed that point. Okay,
It's a tricky one. It's a tricky one.
okay.
All right, we got another question. Tammy's asking about if a sperm and egg could be perfectly fine at conception and then a random mutation happens.
Yes.
Yeah.
So that's the case here. If it happens in cells that affect, I don't know, there's some pretty dramatic pictures of mosaics online. If it happens in a skin cell, do I have a picture of this somewhere? Depending on where it happens and when, I think I have a picture of it somewhere. I may not have chosen to show it here. Here, if it happens right at the beginning when the cells divide into 2, then half of the embryo is going to have the mutation. See that? And here's a baby that is an actual representation of that. That mutation affected the skin. If it happens a little bit later, you can get people who have all these different kinds of patterns, and they do exist. You can find pictures of them online, like a marbled pattern, like those dogs that have marbled fur. You'll find people that can have that marbled pattern because a somatic mutation occurred later in the development of the individual. And then here, this last one is, it happened only affecting the germline. So, the germline is its own compartment of cells in the body. It's different than the somatic cells because it's where cells are made to reproduce the organism in evolution. It's a separate unit of cells, and it undergoes mitotic and meiotic divisions, whereas other parts of the body only undergo this mitotic division. But the mutations can occur anywhere. If they occur in you now, let's say most of your tissues can't regenerate and divide in new cells, but if it did, it would be affecting just 1 or 2 cells and it would never show up. Does that answer your question, Tammy?
Yeah, so either a sperm or an egg was affected for the de novo that Liam has, or it just randomly mutated upon conception. Is that what you're saying?
Yes.
And we'll never know. But
Well, in your case, you had lots of children and only Liam got SCN8A. So I would argue that Liam is a good example of de novo variant, and that none of your other children got it because it happened in 1 sperm or 1 egg. I don't want to blame the dad here.
yeah,
There's no blame to go around, by the way.
No.
It can happen in either side of the mom or dad.
And there really is no way of knowing.
Well, you can test men.
Okay.
Well, if it's a mosaic, you can test individual sperm to see if other variants are in the sperm. That way you can test for mosaicism in a male. You can't test for mosaicism easily in a female because you can't retrieve the eggs easily. But males can be tested for germline mosaicism. As far as de novo goes, you can't tell because it only happened once and the event has come and gone. If there's something else in the DNA nearby the SCN8A mutation that marks the father's DNA versus the mother's DNA, you can do something called linkage analysis to see if it came from the mother or father. But there's no point in knowing because what's the point? What's the point of finding out which parent transmitted it?
mhm. Thank you.
And this is really interesting. Steve put in the chat that Peter has a patch on his side that gets darker when having seizure activity or a fever. That's fascinating.
That is, I don't know what that would be though.
Yeah. And we have a question about what about a parent who tests for SCN8A and their child also got SCN8A, but the parent didn't inherit from their parents?
Does the parent have it too and that is not
I
affected?
believe so. Melina, you can feel free to come off mute and chime in.
So I have the SCN8A mutation. I'm not affected. My son has a SCN8A mutation. He is affected. My mother got tested. She doesn't have it. My father doesn't have it. And then I got my children tested as well, my 2 daughters, and they don't have it.
Sounds like you have it because it was a de novo in one of your parents and you got it, but then you're unaffected. Or you are a mosaic and not all of your cells have it, so you're not affected. But your germline is. See, the thing is, how many children did you have?
I have 3 altogether.
And only 1 got it. So if
Yep.
you are a mosaic, it didn't affect your germline significantly. May only be in a couple % of your eggs. And one of them, I mean, it's possible you would have a child. If you kept having children, if you had 10 children, maybe you'd have one that had it, but it's a very low frequency in your germline. Or you have it, it's not in your germline, it's in all of your cells, but you're protected. I'm going to talk about protection in a minute.
Okay, may I ask the sex of the child that has SCN8A?
My son,
And your other 2 are daughters?
correct.
Dr. Hammer.
It's on chromosome 12, remember?
Okay, yeah, that's fair. Never mind. Never
Yeah.
mind.
Yeah, it's autosomal. It's on chromosome 12. So
And I think, yeah, and it's important to remember that you can have a mutation and not be affected. There are some mutations that are
I'm going to talk about that.
cause disease. Yeah. And then quick one before you move on. Does this question from Sherry about the fact that our case is inherited, does that mean that all children and future generations will have the mutation? I don't know if you're going to hop into that.
Only 50% of the children of your, only 50%. So you have a 50-50 chance of passing it on in an inherited case. Okay, so never is it that all the children will have it. When we talk to our inherited families who've passed this on through multiple generations, we will see that roughly 50% of the children inherit it, and they pass it on at about 50%. That keeps going. Okay. And that's because during meiosis, so even though all your cells have it, only one chromosome has it because it's heterozygous. It's a dominant, autosomal dominant, which means you only need one bad copy, one mutant copy to have the effect of the disease. And in the cells, in meiotic division, one autosome, one from mom or dad, will split into the sperm or egg, and randomly the sperm or the egg will receive either the mutant or the non-mutant chromosome. So I know this is complicated, but the bottom line is, if it's an inherited situation, it's on an autosome, half the children have can inherit it. Roughly flipping a coin each generation. Each generation you flip a coin. Okay? And that's the situation here. So there, up until, I want to talk a little bit about the recurrent risk, in the recurrence risk in future pregnancy. So before 2018, you go to a genetic counselor and they say, well, you had one child with the mutation, your chances of having another child with it are very low. They'll say, and they give you a number like less than 1% or 1 to 2%. But then there was a study in 2018, and it identified that in a group of people who inherited a variant, 10% of them actually had a mosaicism, okay? 1 in 10 of the families had a mosaicism. So therefore, the 1 to 2% risk is not appropriate to give in all cases, especially in the case of a mosaic. So the precise recurrent risk can't actually be given because it could approach as high as 50%, you know, if lots of your gametes had the mutation. So this was a blog written by Katie Helbig on Beyond the Channel archive, and she was saying, we must inform families about the possibility of undetected parental mosaicism and offer a referral to a prenatal genetics professional. And this is getting to Laura's question, to discuss prenatal or pre-implantation genetic testing options. So if you are a mosaic, you'll have to then resort to pre-implantation genetic testing or in some kind of prenatal testing to know if you've passed it on, if you're going to pass it on. Now, there were subsequent studies after this, and this was a study that just came out in 2023, and they figured out a way to tell families what their probability of passing it on was in a more specific way. So they could divide families into subgroups. So let me just read it through here. Following the diagnosis of a pediatric disorder caused by an apparently de novo mutation, a recurrence risk of 1 to 2% is frequently quoted due to the possibility of parental germline mosaicism. But for any specific couple, this figure is usually incorrect. We present a systematic approach to providing individualized recurrence risk. And it's complicated. You have to do this kind of complicated testing by combining locus-specific sequencing of multiple tissues. So they sequence the DNA at a specific gene in multiple tissues, many tissues, to detect occult or hidden mosaicism with a particular kind of long read sequencing to determine the parent of origin of the mutation. We show that we can stratify the majority of couples into one of 7 discrete categories associated with substantially different risks to future offspring. Okay, that's a big mouthful, but the point is, in a research setting, it's possible to subdivide families into one of these 7 groups that have different risks of passing it on. But the bottom line is this. Data demonstrate that for all couples, it's possible to refine the risk of having another affected child with the same de novo mutation. Among 58 families with a single affected offspring, representing 59 de novo mutations and 49 genes, the recurrence risk for 59% was decreased below 0.1%. So it gets, in most families, the risk gets decreased below 1 to 2% to less than 0.1%. For the very few families, the 5 out of 59, 5 or 9%, the risk increases to 5, let's say 6 to 12%. So if you were going to have one of these fancy tests, your 90% of the families would have a risk that reduces to less than 0.1% of passing it on. The unfortunate 10% will have an increased risk of 6 to 12%. So that 1 to 2% is just a ballpark, but 90% have less and 10% have a higher risk. And you wouldn't know unless you did this test. But this is an opportunity to talk to your genetic counselor or your clinician who's in charge of ordering tests and have the discussion about future risk of passing it on. But this is only for the case of de novo germline, okay? In the case of inherited, the risk of passing it on is 50% each generation. All right, so I'm going to go now to looking at family studies and why some people are protected in the case of an inherited variant, okay?
Can we ask, do you mind if we ask a few questions that came up?
Yeah, please do.
Okay. So Gail was asking, are there cases of homozygous mutations? This seems to be a dominant genetic disorder, suggesting most of the cases are heterozygous. What do you?
That's right. That's absolutely right. So if you're talking about autosomal recessive, you're talking about a different situation because you need 2 copies of the mutation to get the disease. In that case, you're going to pass it on to all of your offspring.
Yeah.
Now, you're only going to pass it on in one mutant allele. And if the other parent has 2 normal alleles, then the child will be a heterozygote and it won't show up. They'll be a carrier.
Mhm.
There are some populations in the world that practice something called endogamy, where cousins marry quite frequently. And you'll see a lot of recessive diseases expressed in populations that have cousin mating for that very reason, because you're a carrier and maybe your cousin's a carrier, and you're going to occasionally 25% of the time have a child where they have both copies. That's just normal Mendelian genetics. No mosaicism, nothing. It's just autosomal recessive inherited disorder.
And then Gemma, do you want to ask your question about the twins? Because I think it might be more complex, but sometimes it's hard to come off mute too. Let me find you. All right, I just asked you to
Hi, so obviously the twins have like 2 variants, and obviously me and my husband don't show any symptoms. So obviously, what would that mean for the twins? Obviously,
Remind me again, Gemma.
they display the symptoms, but obviously, what would that mean for like future generations, I suppose? Or like obviously we don't obviously display any symptoms, so
Okay, so we'd have to know. Let's pretend this is this is you, this is your husband, and these are your twins, okay? And the question is, did they inherit both variants from the same parent, or did they get one variant from each parent? Do you know if that's the case?
yeah, one from each parent.
Okay. So, and they're identical twins as far as you know.
Yes.
Right. All right. So you donated one variant, your husband donated the other, and the twins are
Yeah.
identical, so they got both. So now the question is, are they going to pass? So they've gotten one. So on one version of chromosome 12, they have one variant. On the other version of chromosome 12, you got from dad, they have the other variant. And when they have children, they're going to pass on one or the other to the egg. And then some of the children will inherit one, but not both. Okay. They're not going to have both from now on.
Right.
Right? So they'll get one or the other at a 50% probability. And
Right.
what that means, we don't know, because both you and your husband are unaffected, right?
Yeah.
Is it the combination of the two together that led to your daughter's phenotype, if you will, that which we think is loss of function, right?
Yeah, loss of function, yeah.
Yeah, so the two together might be the two mutations together, and this is a very unusual case. There are other families with two mutations in SCN8A, probably like you. One parent had one parent had the other. They're unaffected, but in combination, they cause a loss of function.
Yeah.
I think that's probably the best scenario. And then when they have children, they're only going to pass on one or the other, so their kids are out of the woods, if you will.
Yeah.
Okay, that's a very interesting case of what can happen. So we'll continue to discuss these as we continue on, but let me just bring in one more situation, and that is, here's a family. Now, this is a Dravet syndrome family. It's not really Dravet syndrome, it's SCN1A. There's a variant in this great-grandma. This is a great-grandma. Here are the grandparents, here are the parents, and here are the kids. So with respect to this individual, there's the parental generation, the grandparental generation, the great-grandparents. There is a variant. If you see a shaded circle or square, that individual has the variant. If it's a round, if it's a circle, it's a female. If it's a square, it's a male. So here we have a woman. We don't know how she got the variant. Did she inherit it, or was it a de novo mutation in her parents? We don't know. But it's essentially now an inherited variant, and she passes it on to 3 of her 3 children. Okay, so the coin flip didn't work out so great there. 100% of her children inherited it. 2 daughters and a son. Okay, now they marry somebody from outside the family, so fresh DNA is getting in each generation. And in this family, now in this case, the dot means they got the variant, but they have no epilepsy. Okay, 1, 2, 3, 4 out of 8 kids inherit it. So that's a perfect example of Mendel's rules, 50-50. 4 out of 8 kids got it. In this case, 3 of the 4 don't have any symptoms. Okay, so we're very interested in that case. We'll come back to it in a minute. In this family, 1, 2, 3, 4 out of 6 kids inherited the variant, and they're very mildly affected. And the different patterns in each of these 4 boys shows that they had febrile seizures and atonic seizures, or they had febrile seizures and partial seizures, or they had afebrile seizures, etc. All very mild. In this family here, 1, 2, 3 out of 5 people inherited it. 1 had myoclonic seizures and febrile seizures. 1 had partial and febrile, and 1 had febrile and absent seizures. So this is really instructive. Now, this is SCN1A, but it shows you the same genetic variant that's inherited from generation to generation and is mild enough so everyone does fine. They grow up, they have their seizures are probably controlled, or they outgrow them. They have children and they pass the variant on. That's the subject of today's meeting. And over here, this father who had febrile and absent seizures only married a woman who was neurotypical, presumably, and it passed to both their kids. One of them had very mild effect, And one of them actually had a very severe effect, Dravet syndrome, more extreme than anyone in the family. So this presents us the opportunity to investigate what various factors make the expression of the disease milder or more severe. It also gives us the opportunity to ask, how did different treatments that these individuals in the same family received affect their outcome? Which drugs did they get on? When did they get on them? Those are the kinds of things we can learn from these families, and that I hope we can all pitch in our common wisdom to learn for SCN8A. So let's stop here for a minute and let this sink in, because this is the subject of today's meeting. And I want to make sure everyone understands what the fact pattern is. And then I'm going to talk just a bit about what we found when we did research on this family. Okay, so I think it's pretty clear cut. Everyone gets it. There's a severe case. Most of the people in the family have very mild epilepsy. They can outgrow it or keep it under control. There's 3 people in this generation that don't have any symptoms whatsoever. So why is that? We brought up the case, Lena, about unaffected parents, okay? Maybe this is you. Maybe this is your kids. One of them has SCN8A expressed epilepsy, and maybe, I don't know, I forgot to ask if you had another child, but I think you did, and it doesn't have SCN8A. So why are you protected? Why are these 3 individuals protected? So we did an exome sequencing study of this portion of the family here. And what we found was there was a gene variant that these 3 inherited from this woman here that was not the one that's segregating in the entire family, a different gene variant. And they're the only ones that had it. And when we tested them in a cell system through a collaborator at the University of Indiana, he showed that the variant that they inherited from this woman protected the sodium channel and made it more normal. It behaved more normally. So that's called a modifier gene, a gene other than SCN8A, or in this case 1A, that alters the expression of the mutated gene to make it more normal. It can also go the other way. It could make it more severe. So I would argue that this individual may have received another gene, perhaps from this person, that in combination with the sodium channel mutation made things worse. So this is why you can't say if somebody's unaffected and has the mutation, and they pass it on to an individual that has the mutation and is affected, to say that that mutation is not playing a role in what's happening, which is often the interpretation of the genetic counselor or the clinician. You have to be open-minded to the possibility that there's something more complicated going on, because it happens all the time. It's just that science, or at least medical testing of families like ours, is not progressed to the point where it can easily determine these kinds of things. All right, so that's what I wanted to say. Now I just wanted to say that this slide shows the sodium channel nav 1.6, the thing that's encoded by SCN8A. And I've looked in the literature to find published cases of inherited situations, right? So here's a case where it was inherited and the mutation's here in the N-terminus, and it was in an unaffected mother. And here's the child who has some, we don't know what the effect is. I have to go in and see how severe it is. Here's a case here where one of the parents was a mosaic for 1872Q. Here's a case at 1877S where the father was affected and the child is affected, and so on and so forth. These are all cases that are published in the literature that I'm mining to get all the information I can. I think there's, I don't know, there's a dozen cases here or so. And so I'm in the process of going through this and seeing what the phenotype of every one of these individuals is. And the pedigrees for these individuals, some of them are shown here. So this is an interesting one. Some families might recognize this variant, 1483K or LYS. There are 3 families unrelated to each other that all passed on this variant. And in this particular publication from 2016, they found that the individuals in these families had heterogeneous expression of this variant, but all of them were very mildly affected. Let me just point out that when you have a variant that's affected through multiple generations, the bottom line is it has to be a mild variant because everyone's doing fine and having kids. Here's another one from 2016, 1523D, and you could see it's inherited through multiple generations as well. These are just examples of SCN8A that I showed you, but nobody's done any further research to try and figure out, A, are there protected individuals, and B, how did the different treatments of these individuals lead to different particular outcomes? And here's another set of inherited SCN8A variants. In this family, an unaffected father passed it on to a son who passed it on to a daughter. Here's another case where the father was affected and passed it on to 2 of 3 children. Again, all very mild, controllable seizures, probably. These people grow up and have kids. Here's another case with a different variant, and so on and so forth. So I'm just pointing out that the literature for SCN8A and inheritance and what's going on is still in its infancy, but now we're at the point, as Gabi said, where we can really dig in, and perhaps our community can take us to the next step of understanding a lot about this disease. Here is what we have in the registry, and you can see we have many cases of inheritance, and some of them overlap with the ones I just showed you. But we have affected parents, unaffected parents, we have inherited variants, and a few mosaic. There's a family in Spain where the dad is mosaic and it's at 1617Q, just like Laura was saying about her daughter, but he's a mosaic and her daughter inherited it, his daughter inherited it and is affected. So that's definitely happening. We just don't have many examples where parents have discovered that they're mosaics. So just to wrap all of this up, there's 15 published cases of SCN8A inheritance. There are 29 cases in the registry, 3 that overlap. The phenotypes are always either loss of function or mild gain of function, because these are the kinds of variants that don't affect an individual to prevent them from having children. And so it gets passed on. The recurrence risk for the inherited variants is 50%. And then pedigrees with associated phenotypic data, which is what I want to work on now, are critical to better understand the factors that contribute to mild or more severe disease. We can distinguish nature versus nurture. For example, is it that the family members were on different drugs at different times after their first seizure? Or is it just genetic background that's dictating different levels of severity? And finally, this is so important. By documenting all of this in our families, future generations will benefit because they'll have a record of the family history, and information will be able to be presented to clinicians on treatment and the importance of testing other family members. So when you argue with your doctor, I want to know, since it's inherited, who else in my family has it, because they need to know. And I need to get them on drugs like I had when I was a kid. This research will support that process. So I want to end with that, and I want to open it up to the conversation that families can share as much as they want about their own situations, the struggles they've had in getting tested or getting medications, because these are mild. And so doctors or clinicians may say, well, you don't need to go on a medicine. Well, I know 3 other people in my family, all of them have needed medicines. Yes, my son's young or my daughter's young. They're not having their first seizure yet because it may come later. So I still want to discuss the idea of getting on a medicine as a preventative of that first seizure. All right, before I turn the slides off, I'm going to ask any more questions. I can always come back to them later, but that's all I wanted to say.